cooperate and how those cooperative relationships are maintained, especially over long periods of time. So a few different things that partners can do for each other is provide each other food, housing, or protection from predators. And these are, of course, classic examples of symbiotic relationships between lichen, acacia ants and their trees, and fish. Another exemplary symbiosis is that of leafcutter ants. And of course, these ants grow and consume fungus. The leafcutter ants use fresh leaves, but ants in the tribe Actinidae, which these leafcutters are the most derived taxon, they can also use leaf litter and other substances to grow their fungus. And the important thing about these ants is that they very closely co-evolved with their fungus over the years because their fungus is entirely dependent on the ants for its own propagation. And so what makes this particular symbiosis interesting, in addition to the ones that I previously mentioned, is that because these ants are farmers, they can provide us with a lot of information about how disease control can be used as a mechanism for maintaining symbiotic relationships. Because these ants not only have the relationship with their fungal cultivar, but they also have a problem. So they have pathogens such as Escabopsis, which is a pathogenic fungus that infects their fungal cultivar, which in turn, of course, impacts the ants because this is their food resource. And so in turn, the ants have co-evolved with a bacterium called Actinomyces, which produces an antibiotic substance that we apply to the fungal cultivar, and that combats Escabopsis infection. So within this general schematic of this complex evolutionary relationship of disease control, the fungal cultivar seems to be somewhat passive, but in reality, the fungal cultivar does a lot for the ants besides provide it with food. So it also provides a structure within which the ants store their brood. So if you look underground at this particular species and open up one of their garden structures, you'll find ants running around with eggs, larvae, and pupae in their mandibles. And if you take a close-up look at one of these larvae, for example, you'll find tiny little patches of mycelia deposited on the larvae or the pupae or the eggs. And this mycelial cover has been hypothesized to serve a number of functions, but one of the major ones is that of disease control, so protecting the brood from microbial pathogens. And of course, this makes sense in the larger context of this evolutionary symbiosis because the fungus relies on the next generation of ants to bring it to new colonies so that it can grow into new gardens. And one interesting thing that we see about this mycelial cover on the brood is that across the taxa, we see different patterns of how much mycelial cover are on broods of different stage. So in the lower and the actines as well as the leafcutter most derived, we see this pattern where younger brood have more mycelial cover, or less mycelial cover than older brood. So same in the leafcutters. However, in these transitional taxa, we see the opposite pattern, where younger brood have more mycelial cover than older brood. This is a little counterintuitive if we're thinking of mycelial cover as a mechanism for disease control, because one might expect the degree in which workers use mycelial cover to vary across taxa, but not necessarily the direction. So you might think that all larvae are universally more vulnerable to microbial pathogens than pupae, or vice versa. So this is an interesting phenomenon that we see in the field data, which has been vastly oversimplified by me, but it's something that hasn't been investigated yet. And it's also something where we don't know whether this pattern is something that's a product of the environments in the field, so predation, or if it's actual worker deposition of mycelial cover, so if it's based on behavior. So to investigate this, I looked at Hermogenes Fernandez, which is exemplifies this pattern of younger brood having more mycelial cover than older brood. And I wanted to ask, using Fernandez, how do workers allocate mycelial cover based on brood type? I also wanted to know what role the garden structure plays in how they treat brood of different stage, because this has been largely unexplored, and yet this is basically structure into their own small garden. And I gave them one larva and one pupa each to compare their responses to brood of different stage. And so what I did with that brood was, before I placed them in the samples, I photographed them. Then I photographed them afterwards. So in image J, I compared brood photos before and after the trials to see what kind of different levels of mycelial cover I had and how that changed over the duration of the 3-day trial. I also took measures of where the workers placed the brood in the garden. So one of them was height, so whether the brood were closer to the top, middle, and bottom of the garden. And the other one was contact, so basically on a scale of 1 to 5, how embedded the larvae or pupae were in the garden. And something that was interesting that resulted from my samples was that most of my samples I put a larvae in and at the end I still had a larvae. Some of them, of course, larvae transitioned into pupae, so some of the larvae were slightly older and had become pre-pupae by the end of the trial. Some of the larvae had become new pupae by the end of the trial. So rather than throwing out this data, I wanted to look at how this continuum of larval development could inform what we understand about worker deposition of mycelial cover on brood of different stage. So this will be the format for the next couple of slides. I'll have 3 figures showing you the different sample types. So the youngest larvae that were still larvae at the end of the trial, the larvae that had become pre-pupae, so that were in morphological transition to become pupae, and the samples where it had 2 pupae at the end of the trial, one of the old pupae that I deposited, and the other very new pupae that had originally been a larvae. So larvae, original larvae on the left, original pupae on the right. So for the main samples, what I found was that predictably, according to the field data, larvae had a greater deposition of mycelial cover than pupae in the samples. So younger brood were getting more mycelial cover over the duration of the trial than pupae. However, that breaks down once the larvae start transitioning morphologically into pupae. And you can see a very tiny downward trend, negative trend. And then this is exaggerated in the duration of the trial when the larvae have become pupae by the end of the trial. So you see a negative trend in the deposition of mycelial cover. And this makes sense if we see across the population that larvae have more mycelial cover than pupae. At some point, that has to decline. So one of the other measures I took was garden depth in terms of worker placement. And for samples where we had 1 larva and 1 pupa at the end of the trial, we see a similar pattern of worker placement for larvae and pupae. So most brood, generally speaking, are in the middle of the garden, and some are at the bottom. Whereas this, we see a very different relationship between the 2 different brood types when it comes to pre-pupae and pupae samples. So it seems as if for pupae, it doesn't matter too much where the workers place them in the garden. They're generally distributed at the top, middle, and bottom of the garden, starting along the vertical column, whereas larvae are still mainly in the middle and even less at the bottom of the column. And once we get to pupae, that relationship seems to collapse a little bit. So it seems as if at the beginning and at the end of the trial, the youngest larvae and the oldest larvae are actually being treated similarly to the pupae. And when we looked at garden contact, we saw something different. So here, black is larvae, gray bars are pupae. And so 0 is the least amount of contact, basically no contact with the garden, and 5 is almost completely embedded in the garden. So we saw generally that there were moderate levels of garden contact for larvae, whereas a fairly even distribution for pupae. And so you see pupae with almost complete envelopment in the garden, but no larvae at that stage. And then when larvae were transitioning morphologically, it seems as if there's even a little bit less contact with the garden, and pupae look pretty much the same. And when larvae have become pupae, or very recently become pupae, you see that the trend starts to match that of the pupae. So to explain the last couple sets of slides, it seems to me as if larvae are moved into the middle of the garden during morphological change, as we saw in the last slide. And this in turn breaks contact garden, they may have less access to them and are depositing less. No problem. So to summarize, we did see what the field data suggested, which was workers do deposit more mycelial cover on younger brood in this particular species. And larvae at the morphological transition seem to be repositioned within the garden structure, and that's something that requires further investigation. And so that implies that these workers are working at a very fine time scale in terms of larval development and how they treat larvae and pupae at different instars. And it also in general implies that this contradictory pattern that we see across the phylogeny shows different measures of how workers treat brood at different stages in terms of disease control. So this definitely merits a further cross-taxa investigation to understand this particular reverse phenomena of mycelial cover deposition, because that could help us understand how disease control functions in the larger arena as a mechanism for maintaining this important symbiosis. So I'd like to acknowledge my funding for this internship at the Smithsonian Tropical Research Institute. My advisors in Panama, William Wcislo and Hermogenes Fernandez, as well as Ernesto Bonadillas, who helped me dig up a lot of ants. And also, before I get in trouble for copyright infringement, I should mention the photos that I used earlier for Cleaner Fish was by Krillfish on Tumblr, Lichen, Umberto Salvignon. Acacia ants was by Murtaza Handiwala, and leaf cutters were photographed